Well, now's as good a time as any. We'll take her down to periscope then.
Yes, sir. Clear the bridge. Dive.
Oh, my mic means muted. Greetings, everybody. This is Salvador Cordova. Welcome to the Evidence and Reasons YouTube video channel. This is a channel devoted to exploring evidence and reasons for the Christian faith. I'm privileged to be joined by Otangelo Grasso, and he debated Dapper Dino yesterday on the topic of
irreducible
complexity and evolution. So I felt it'd be appropriate to explore this paper, Genomics and the Irreducible Nature of Eukaryotic Cells. So what Journal Club is, when I said nerds go clubbing, that's the idea, that's a play on words of where people say night clubbing. And in this case, Nerds Go Clubbing is referring to what they call journal clubs, that professional researchers occasionally have gatherings where they go through peer-reviewed literature together and try to read through it and just try to learn. Often it usually is up-to-date, the most up-to-date or cutting-edge papers, but there's no restriction that on just studying those. So this is an older paper that was around 2006, and it would be good to compare and contrast with the present way, the mainstream views. So anyway, we're just going to read this paragraph by paragraph and then pause and break for commentary. I may occasionally have to take a commercial break to take care of some other things. And Otangelo, you might have to just fill in, and because I'm the one who has to kind of scroll through this. So Otangelo, let me take a commercial break for the moment. Please go ahead and just share some thoughts on your debate yesterday.
Well, I think it was difficult because they brought up examples of irreducible complexity on which I didn't study before. So I'm doing that right now, and well, we'll see. I don't think that they will have really a case. I'm skeptic that the cases which they mentioned, which is lizard evolution and placenta and so forth, that this is really something that will demonstrate that non-functional parts evolved to then become functional and basically falsify irreducible complexity. But besides this, I think it was a good debate. It has provoked a lot of exchange and people commenting at the comment section of the videos. And there was also the after show, which was nice. I think Deborah had a good moderation skills to permit that everyone of the people had their talk, including me. And it was a good time. It was fun. So I think that it was productive, and I hope that it can spur more conversations amongst both parties. So I think it is good that we talk about these things, and that each side can bring up their side, and that this can incentivize people to investigate, to think about these issues, explore more, learn more things, and so grow in their understanding of scientific evidence and where it leads to. So I think it was a great time, and I hope that such debates can be had more frequently. And I think the Sal's channel here is a good place to investigate molecular biology, proteins, abiogenesis, whatever comes up in regards of origins, where it leads to. And I think we can learn a lot here, and I hope that I can bring some participation and some things to the table too.
Okay, I might have to take a commercial break after a couple paragraphs, but we'll just go through this. And I'm gonna try to improve the Zoom so it can be a little bit clearer for our audience. So, genomics and the irreducible nature of eukaryote cells by Kurland, Collins, and Penny. Large-scale comparative genomics in harness with proteomics has substantiated fundamental features of eukaryote cellular evolution. The evolutionary trajectory of modern eukaryotes is distinct from that of prokaryotes. Data from many sources give no direct evidence that eukaryotes evolved by genome fusion between archaea and bacteria. Comparative genomics shows that under certain ecological settings, sequence loss and cellular simplification are common modes of evolution. Subcellular architecture of eukaryote cells is in part a physical-chemical consequence of molecular crowding. Subcellular compartmentation with specialized proteomes is required for efficient functioning of proteins. Now, the thing I got out of this is you'll hear conflicting views on all of this. Some people will say, yes, there's fusion, and others will say, no, this was the no. So I just want to point that out. What I do point out is when evolutionary biologists disagree with each other, they as a matter of principle, at least at best, only one of them is right. The rest of them are wrong. Does it change how we do biology? Which one we pick? That just means to me that evolutionary biology is pretty much useless to understanding how biology really works, because you'll read one textbook that'll say fusion and others that don't. And I'm like, well, what does it matter? That doesn't change a thing. So anyway,
what do you do? What do they even mean with fusion? What is that?
Maybe that's something we'll learn along the way, and maybe we file that away. So I'll continue here. Comparative genomics and proteomics have strengthened the view that modern eukaryote and prokaryote cells have long followed separate evolutionary trajectories. Because their cells appear simpler, prokaryotes have traditionally been considered ancestors of eukaryotes. Nevertheless, comparative genomics has confirmed a lesson from paleontology. Evolution does not proceed monotonically from similar to more complex. Here we review recent data from proteomics and genome sequences suggesting that eukaryotes are a unique primordial lineage.
Yeah,
I mean, what do we do with that?
That kills common ancestry. And there are even science papers which say that bacteria and eukaryotes are two different things, very explicitly.
And we had another journal club meeting where maybe a couple of weeks ago, Emery and I covered that they don't even agree that there's a last universal common ancestor, single organism for eukaryotes. And they kind of left it ambiguous about the first, what they call FECA, first eukaryotic common ancestor. And now this one's saying could be a separate lineage. What do we make of that? The point is, what difference does it make that we adopt? It doesn't change the way we do biology. And my point is, we could just dispense with evolutionary theory. It doesn't help us understand operational biology.
Well, the thing is, if you have to explain just one last universal common ancestor, that is already difficult enough. But if you have to actually explain a population or already the start of different lineages, that is much more difficult if you assume evolutionary origins.
Yes. So let's continue. Mitochondria, mitosomes, and
what are mitosomes?
A good question. Okay, this is why it's good to have a journal club, because we can look it up. And I think it's just great. We can let me Google it. And I'm embarrassed. I probably studied this sometime in cell biology, and it just, you know how it is. You take an exam, and it's like I forgot about it. So this is why it's good to have a journal club. A mitosome is an organelle found in some unicellular eukaryotic organisms, like members of the supergroup Excavata. The mitosome was found and named in 1999. Okay, so this is recent.
Mm-hmm.
And its function has not been yet well characterized. Can you Google on that and read it? I need a little commercial break, Otangelo. Can you?
Yeah.
And if you want to talk about mitochondria, that'll help too.
Okay, I can just go on here and
I'll let you share your screen, and you can take over for a little bit and just share screens, mitochondria, mitosomes.
Okay, I'll figure that out here.
All right, you have the screen, and I'll join you shortly.
Okay. Mitosomes are almost certainly derived from mitochondria. Like mitochondria, they have a double membrane, and most proteins are delivered to them by a targeting sequence of amino acids. The targeting sequence is similar to that used for mitochondria, and true mitochondrial pre-sequences will deliver proteins to mitosomes. A number of proteins associated with mitosomes have been shown to be closely related to those of mitochondria, or hydrogenosomes. which are also degenerate mitochondria. Current knowledge indicates mitosomes probably play a role in iron sulfur cluster assembly, since they do not display any of the proteins involved in other major mitochondrial functions, aerobic respiration, and biosynthesis, and they do display proteins required for iron sulfur cluster biosynthesis, like frataxin, cysteine, desulfurase, ISO1, and the mitochondrial ASP70. Okay.
Okay, I've rejoined. Thank you, everyone who's joining us. So I'm going to bring back up the paper. So we paused a little bit for mitosomes. hydrogenosomes. So now that's another term.
Yeah.
Otangelo, would you like to try to cover that?
Well, the thing is that I cannot share my screen without you permitting it.
Yeah, go ahead. We'll alternate. So if you could.
Yeah, that's the thing, that I am not able,
if
unless
you just share your, because it looked like you're able to at least get one screen.
What is the name of that other thing that we just mentioned? Let me check here.
Hydrogenosomes. Yeah, I'll show it to you on the screen. Hydrogenosomes.
Okay. I was thinking, just if you have other things to do, I could watch and share the video of Stairway to Life. What do you think?
No, that's okay. We'll go through this. Things are a little more stable now.
Okay. So do you want to continue?
Okay, were you able to find hydrogenosomes? Anything on hydrogenosomes?
Just a moment. Hydro.
Okay.
Yeah, I got it here. Let me check if I can. So here you have to put it on the screen. I cannot do it. Yeah.
There you go.
Okay.
A hydrogenosome is a membrane enclosed organelle of some anaerobic ciliates, fungi, and animals.
Mm-hmm.
Do you want to read it out?
Yeah, the hydrogenosomes of trichomonads, the most studied of the hydrogenosome containing microorganisms, produce molecular hydrogen, acetate, carbon dioxide, and ATP by the combined action of pyruvate, ferredoxine, oxidoreductase, hydrogenase, acetate succinate, CoA, transferase, succinate thiokinase, superoxide dismutase, malate dehydrogenase, ferredoxine adenylate kinase, and NADH, ferredoxine oxidoreductase, are also localized in the hydrogenosome. It is nearly universally accepted that hydrogenosomes evolved from mitochondria. In 2010, scientists reported their discovery of the first known anaerobic metazoans with hydrogenosome-like organelles.
Sounds like another story to me, but this is actually kind of interesting that each eukaryote may have specialized organelles that other eukaryotes don't. So
I have made a long list, which I think is far from complete, about all the different kind of organelles that are contained in eukaryotes. And we have split of plant eukaryotes, of animal eukaryotes, of unicellular eukaryotes, so various kinds of eukaryotes. So I think that the transition from prokaryotes to eukaryotes would have been enormous, and the diversification as well. So I don't see how that would be possible by evolutionary means.
Yeah, so they have chloroplasts in plants. I don't know of any animal that has it. I thought they had some animal that had some things, but anyway, I'm going to continue with the paper. I'm going to go to this figure 1 diagram here. So they have here cellular life, and then all the supposed common ancestors, these phagotrophs, autotrophs, saprotrophs, heterotrophs.
Saprotrophs.
Do you want to look that one up?
Yeah.
No, this is actually educational because when they talk about some of these other, I'm always surprised they find these other life forms that are really very different. Like I heard of something called a chemotroph, and it's like, okay, how does one evolve from the other, and how do they have common ancestors? And we don't have very good understanding of some of these others. So you've not heard of saprotrophs before?
It is kind of a fungi. How do you say? Fungi?
Fungi?
Fungi, yeah. And soil bacteria. Saprotrophic microscopic fungi are sometimes called saprobes. Saprotrophic plants or bacterial flora are called saprophytes.
Oh, it just occurred to me. A chemotroph is a prokaryote. Can I look that up?
Here.
I'm going to embarrass myself here. So this is why I like Journal Club, is it gives a chance to kind of review understanding of things. So chemotroph. So I'm going to look that up, the chemotroph. All right, so I just relearned something. Chemotroph are organisms that obtain energy by the oxidation of electron donors. So chemotrophs can be either autotrophic or heterotrophic. It doesn't say whether they're prokaryote or not. Are they eukaryotic?
I suspect these are bacteria.
Okay. Most chemotrophs are extremophiles, bacteria or archaea that live in hostile environments. Oh boy. Now I'm embarrassed. This is a whole... So they have autotroph. And let me look at Kurland and Penny's. They have autotrophs. So the autotrophs are probably prokaryotes. And you said these saprotrophs are eukaryotes. So already there's kind of a mix right there in the common ancestor. So they're mixing prokaryotes and eukaryotes as the common ancestor.
Yeah.
Oh well. So let's look at the figure caption here. Oh boy. I'm having a hard time navigating here. I apologize. The common ancestor of eukaryotes, bacteria, and archaea may have been a community of organisms containing the following: autotrophs that produced organic compounds from CO2 either photosynthetically or by inorganic chemical reactions; heterotrophs that obtain organics by leakage from other organisms; saprotrophs that absorb nutrients from decaying organisms; and phagotrophs that were sufficiently complex to envelop and digest prey. Endosymbiosis of mitochondrial ancestor. So this is where they're hypothesizing endosymbiosis. So this is interesting. They're almost suggesting that the eukaryote had everything in it, and then they had a that's when they had endosymbiosis after the eukaryote.
I've seen that argument, Sal, that they say that the pre-Luca organism was actually even more complex than today's eukaryotes, and that then they basically split into eukaryotes, which were simpler, and bacteria, and that precursor had both the organelles and so forth, the parts of bacteria and eukaryotes incorporated. So you would have an origin of life of an organism which was more complex than bacteria and eukaryotes, everything mixed together, and that it would devolve to become then specialized to become the three domains of life.
And that echoes what happened when Emery and Moina and I were talking about eukaryotic evolution last week. Oh, by the way, greetings to Hate, Love, Nothing and Doki Doki. Thank you for joining our little journal club this evening. This is going to be definitely a nerd talk. So let me share Otangelo's screen. There you go. Thank you, Otangelo. The sapotrophic nutrition. Yeah, see, this is just enlightening to see how they're invoking all these different scenarios. They don't know, they can't reconstruct. And the point is, it doesn't matter. It doesn't matter how they try to reconstruct it. It doesn't change the way we understand operational biology. And just repeating myself, this shows this is totally worthless. It doesn't add anything to our understanding. It just spams the scientific literature.
It's basically just hypothesis over hypothesis, made up stories, one after the other, and each one of them they see is not satisfying. It doesn't provide a compelling answer, and they just go on with one hypothesis after the other. I mean, we have over 40 different hypotheses in regards of the origin of life. And then look up how many different variations there are to explain the transition from prokaryotes to eukaryotes. There are so many, and none of them is satisfying because we are simply talking about different things here.
Yeah, and it's like it doesn't do anyone any good, really, except it just does the naturalistic community good. It doesn't really advance scientific understanding one iota.
No.
So anyway, let's see. I'm going to re-add this back to the stream. And so, by the way, leave your screen share up too, because then I can. Do you have dual monitors, Otangelo?
No.
No. Okay. It makes it really easy for me because I can actually see what's on my screen, and then, and anyway, we'll just move forward. So that was a lot just to get through one paragraph. I learned about hydrogenosomes and mitosomes. Recent analysis also suggested that early eukaryotes had many introns, and RNA and proteins found in modern spliceosomes.
That is a big issue. How do you explain the origin of the spliceosome and the introns and exons? And you need all the signaling and to know where to split. So how did all that kind of new complexity emerge from when it wasn't there yet? And I think this is also a prime example where you need everything together. That is the spliceosome, the splicing code. You need the genome already with the differentiation of exons and introns. You need information to know where to split, how to rejoin, and so forth. How do you get all this?
All right, we'll just continue. Indeed, it seems that life history parameters affect intron numbers. In addition, molecular crowding is now recognized as an important physical chemical factor contributing to the compartmentation of even the earliest eukaryote cells. Nuclei, nucleoli, Golgi apparatus, centrioles, and endoplasmic reticulum are examples of cellular signature structures that distinguish eukaryote cells from archaea and bacteria. Comparative genomics aided by proteomics of CSSs, such as mitochondria, nucleoli, and spliceosomes, reveals hundreds of proteins with no orthologs evident in the genomes of prokaryotes. These are the eukaryotic signature proteins. I want to pause just a bit. So let me see. There's a diagram. I saw it earlier. There's a diagram. Let me zoom out, and they're going to show the parts of the eukaryote, and I'll zoom in on that. Here it is, and I can zoom in. There was one thing that was interesting, and I need to go back. Otangelo, can you look up the word ortholog? I know there's a distinction. I use the word homolog and paralog so much, I've actually somewhat forgotten ortholog is.
Okay, let me check that out.
So in the meantime, I'll show this diagram. So the ESPs,
Morphology. Orthologs are genes in different species that evolved from a common ancestral gene by speciation, and in general, orthologs retain the same function during the course of evolution. Identification of orthologs is a critical process for reliable prediction of gene function in newly sequenced genomes.
oh boy, I didn't think that that definition was very helpful.
That's in Science Direct. Sequence homology.
Let me look at ortholog. So, guys, this is how we learn science. We sometimes just...
That's a better definition here. Here, orthologs are defined as homologs in different species that catalyze the same reaction. Is that clear?
Can you read it again?
Just a moment. Orthologous genes are genes in different species that originated by vertical descent. No, that was not right. Ortholog identification are defined as homologs in different species that catalyze the same reaction.
So I think I have a helpful diagram. Actually, Wikipedia has a nice diagram for this. So a paralog, I understand that. That's because a paralog happens when it's a duplication and it's in the same. So like topoisomerase alpha and topoisomerase beta, the humans, those are paralogs. They happened inside the same, I think they're paralogs through gene duplication. Now orthologs, either a speciation event or a duplication. Oh boy, see, I totally messed that up. So we have the supposed human and chimp, human histones. Okay, a speciation event produces orthologs in the two daughter species. So what was your definition again that you liked? I feel really bad now. I should know this. Oh my goodness, I'm embarrassed.
Orthologs are defined as homologs in different species that catalyze the same reaction.
Orthologs. So you said orthologs are homologs. Is that correct?
Let me check here. I opened the site here. Let me check here. Oh. Here, orthologs are defined as homologs in different species that catalyze the same reaction.
Okay. So an ortholog is a homolog. Okay, it's a subset. And I think a paralog is within the same species where the split happened. I'm almost sure of it. So let me look. Paralog.
And here in Wikipedia, it says orthologs or orthologous genes are genes in different species that originated by vertical descent from a single gene of the last common ancestor.
Okay. Now this is clearer. I've used the term paralog a lot. So if they suppose that in one species, say a human or whatever, that the gene duplicated in that species and then it got inherited, then those similar genes are then called paralogs.
Mm-hmm.
If it's split because of speciation, it's called an ortholog. So both paralogs and orthologs are homologs. Okay, I'm really this was already worth it. I mean, this is how we learn how to read through Journal Club and read through journals. It's like, oh my goodness, I don't quite remember what that term means. Okay, so yeah. All right. So where were we? So we were talking about CSSs and ESPs. Okay, an ESP is a eukaryotic protein. All right.
Let me just address this stuff I watch sometimes. He asks here, wouldn't endosymbiosis of mitochondria require cells that previously run on something other than ATP? No, as far as I understand, bacteria, they also run on ATP. So,
okay, so stuff I watch sometimes, that's a very good question. How did the eukaryote without mitochondria survive? I would suppose there are other pathways, like I think, like that include the glycolysis cycle that can create ATP without going through ATP synthase. I should know this. So
yes, but the yield and production of ATP through glycolysis is small.
Yeah.
So I don't know if that would be sufficient to drive the bacteria.
I should know this because I took biochemistry and now I'm having to
ATP synthase is claimed to have been existent in LUCA, in the last universal common ancestor.
Glycolysis. I'm sorry, can you repeat that again, Otangelo?
ATP synthase is claimed to be extant in the last universal common ancestor. So I don't know of any living being which can exist without ATP synthase.
That's a really good. Okay, so that is an opening. That is a open question. Are there any creatures that can live without the ATP synthase? We know that there are other, as you said. Okay, here. So there is one where we can have glucose that creates ATP through glycolysis, and this is the ATP synthase. This one here, the chemiosmosis. However I pronounce it, chemiosmosis and electron transport chain.
Mm-hmm.
That's the ATP synthase. There is the Krebs cycle, and then
that's aerobic respiration, and the ultimate oxygen acceptor in the chain. So that's why it must be aerobic. But anaerobic respiration.
Let's see if they have.
I don't remember now if it requires ATP synthase as well.
Wow. Well, we can take a quick look at anaerobic respiration. See if they have a diagram. But thank you for putting that out there. Stuff I watch sometimes. How could a eukaryote survive without?
Yeah, because for what I understand is the alternative to respiration with ATP synthase is fermentation. Now, I'm not sure if fermentation is enough to drive bacteria.
Well, we're not going to get through the paper at this pace. And I mean, if we need to stop that, we'll stop. But that was a really good point. How did, if they're invoking, as this paper suggests, that endosymbiosis happened after eukaryotes were already created, that would mean that the other organelles, I mean, there are different scenarios where you had the nucleus after the endosymbiotic event or the nucleus before. And without that mitochondria, how did it live? That's a really good question. So the reason this is good is we can pose that in our problems with eukaryotic evolution. Thank you very much, Stuff I Watch Sometimes. Wow. So eukaryote proteins. So those eukaryote signature proteins. All right, so let's just continue. I'm here, if you can see my cursor. For yeast, the largest protein class contains more than 200 eukaryote proteins, ESPs, targeted to the mitochondria, but encoded in the nucleus. that's another thing we need to go through, because let me show you something. If you have a mitochondria here and you have 200 proteins here, and this is just in yeast, that means you need localization signals, one for the RNA, just to get it out of the nucleus. So that's one level. That's one level. The next...
I didn't know that the spliceosome is inside of the nucleus.
Yeah, this is why it's good to go through Journal Club, because even when you're taking through classes, the professor has his or her set of questions, and you kind of just learn that. But then there are other things that were probably in the textbook that you just kind of skipped. So this is really good. So the spliceosome is inside. Now, I need to tell you something that's interesting. And this is also true. I showed it with the RPS3 protein. Okay, how do we build the spliceosome? Well, first, it has to create all of these RNAs. The RNAs need an export signal. The export signal will take the RNA, the mRNA out here, and then it's going to find a ribosome. It's going to get translated. After it gets translated, guess what?
It goes into again to
it's
the
an import signal to get here, and
it goes traveling.
Right. I'm just saying, this is how I want to. You know, we had the creationists rightly were focusing on abiogenesis. The next step is, okay, now this is an evolutionary scenario where we're talking about the transition from prokaryote to eukaryote. I said, take it whichever way you want. You want to start out with prokaryotes, with eukaryotes first. I said, okay, that's kind of funny, you know, where it devolves, and that's just as ridiculous as evolving. So, you know, take it either way. It's going to be bad for the evolutionists. But you see the problem. I'm planning to try to hammer this the whole rest of the year. I had a conversation with our dear Dr. Sanford. Praise God that his angioplasty went well. And he announced that in his talk with James Tour. That's not private information anymore. And I told him, I said, Dr. Sanford, this problem with the import-export, it's going to be devastating to argue for universal common ancestry. You'll have to invoke miracles. You see exactly the problem here, Otangelo.
I have studied this, Sal, and I have a site here at my library, and there are import channels. I think they are, the name is quite funny. I think it's Tip Top or something like that. I can look it up afterwards. But that issue here that you require to import and export, I've seen that a long time ago, and that is one of the reasons that I rejected endosymbiotic theory, because you need to create all these channels, and they are hyper complex.
They're hyper complex, and then you have to. It's not just building the channel. You can't. You know, they can't just take some phylogenetic reconstruction and say, "Oh, that's clear, it evolved," and like that's not an explanation mechanistically. So when we have these orthologs that are unique, and then even if they're homologs that exist in prokaryotes, how did it get the import-export signals? It needs an export signal just at the RNA level, and then an import signal to bring it back. And in the case of the ribosome, some of the subunits go through several rounds in and out of the nucleus.
Okay, can I put this here quickly on the screen?
Sure, go ahead.
You'll be fascinated by seeing the complexity of this thing here. It's crazy.
All right, I see it coming up. You have the control, sir.
Yeah, here in the chloroplast, you see all that, the tick, the talk. This is all import and export channels here.
In a chloroplast, it has import. I didn't know that.
Yeah, here.
Oh, and you're going to love this. Since you love ATP synthase, hang on. I'm going to show something. Yeah, you go ahead and explain that if you want. If you'd like.
No, because when this was in 2013 that I collected this information 7 years ago.
Synthase. This whole thing with import-export is going to be a big deal, because all these evolutionists do is just do a phylogenetic reconstruction. They don't actually think. It's kind of a brainless process. and it does adds no insights. It's not critical
I
thinking
mean,
at all.
just by the number of proteins which you see in the pictures here, you can imagine the quantity of information processing that must go on through these proteins just to direct the action. That must be crazy.
Yeah, we're going to have a lot of fun exploring this, the topic of import-export. And so what was the name of that? Now I'm going to show, this isn't really a very good picture, and I had a better one. Let's see if I could find one. I was going to show you the ATP synthase. This really stood out. I mean, you probably already knew this, but I remember looking through some diagrams. Okay. So all right. This is actually more clear here. In a mitochondria, you see how the ATP synthase, you see the direction it's facing?
Mm-hmm.
In the chloroplast, it's reversed.
Yeah. Yeah.
I mean, how did that just flip? I mean, you don't just flip a molecular machine like that.
Yeah.
And I remember looking through in my biochem book, and I said, there's something that doesn't jive. And then I finally found this one diagram that actually compares it. You don't notice it when you just look at it in isolation, and then you realize, oh my goodness, the things are oriented differently in the membrane. I mean, did it just one day decide to just kind of flip the direction of the ATP synthase?
Yeah. Yeah.
Oh my goodness, we're going to have so much fun with this. This is why, God bless the Standing for Truth channel, but this is why I really enjoy talking about eukaryote, prokaryote evolution. You just have so much of this, and the other side just, they won't even show up. Oh my goodness, they won't even show up in debate. If I started debating stuff like this, there's not going to be any opponent that would want to even touch it. Once he sees me, if they see our conversation like tonight, I'll tell you this. I wrote Speedus out. I said, you want to debate eukaryotic evolution? He was saying, I'm going to kick your butt. I said, okay. And I wrote him. He never got back. He just had all these weather reports he keeps redoing. I was like, okay, so you want to spend your time with that. You don't want really to engage. He said, I'm going to kick your butt on nuclear pore complex evolution. I said, okay.
Well, Sal, what I've observed, and I've seen that many times happening, and I am sure that he will listen to what I am saying now, and then he will criticize me, but it's exactly that. When I bring up something which is very clearly pointing to design, he moves on to another topic. He doesn't go even into it.
Yeah.
You know?
And then he'll say, look at all the papers that solve this. And I'm like, bull. They're just phylogenetic reconstructions. They're not mechanical explanations where we have just tried to say, okay, you can't have a random, even if you assume that your phylogenetic reconstruction might show a common ancestry path, those mutations can't be random because the creature would be dead. It'd be dead. I mean, how are you going to build a spliceosome when its parts are going in and out?
Well, I think the overarching problem here, Sal, is the information. Everything that changes requires new instructions in order to be able to change. It doesn't happen chaotically and randomly and without direction.
Oh, Doki pointed out something. RJ scared to debate eukaryote evolution. He was going on my channel and just talking trash. I said, I'll debate you on eukaryote because he was saying you don't know anything. I said, okay, why don't you debate me on eukaryote evolution?
RJ, is that Roland James?
Yeah.
Yeah. No, he's close-minded. I mean, I'm having a continuous exchange with him per email, and I mean, I've provided so much evidence about my points, and he simply ignores it, so.
Okay, so if we don't finish the paper, we can, but while we're having fun, we'll keep going. So so I think now I really want to do this nuclear pore complex thing. It's not just the pore complex. It's also all of the localization signals. There are at least 2 levels of localization signals for things that are nuclear proteins. You have to have the RNA export, and then after it codes, it has to have a protein import signal. So it has to be coded in the RNA and then also in the protein, and they're 2 separate signals. I'm just
now,
like,
Sal, does science know these signals, how they are generated, and how that works?
we know how the signals are read a little bit for the protein side. If anyone knows, they're welcome to ask about the import-export of RNAs. I don't think that's well characterized. The Maverick asked, can I ask a question that may sound silly? How can we say single-cell organisms are not the result of decomposition or of a larger organism with eukaryotic cells? Otangelo, I think you covered that. You can share that with Maverick, and I'd like to thank everyone who's joining joining us in our as we go clubbing here. So,
with decomposition, what does he mean by that?
Like
devolution or
devolution? Yeah, devolution.
Well, the thing is that
this before, and if you could just repeat it again for him.
Well, the thing is that I can give you a link, and you will see the huge differences. The cell membrane of prokaryotes and eukaryotes is different. The ribosome is more complex in eukaryotic cells. The DNA replication is different, and there are many other things which simply are not matching. And you cannot say that one derives from the other. That simply doesn't work. And that goes to a point that science papers, they very openly admit that the common ancestry of bacteria with eukaryotes, that simply is not true. That doesn't work.
And I would just add this: if everything devolved from a more complex organism, that's genetic entropy. And that's really kind of funny that to rescue evolutionary theory, you have to appeal to genetic entropy.
Yeah. No, but I think a huge problem, Sal, is also following that they claim that the first carbon fixation mechanism was either the Leung-Wutthal cycle or the reverse TCA cycle. Now, that is for an anaerobic organism, because they claim that the first life did not, oxygen wasn't there yet. Oxygen that was produced with cyanobacteria about a billion years later. So now the problem here is that first you have a cycle which operates anaerobically in an environment without oxygen, and then suddenly 6 different carbon fixation cycles emerged somehow with different protein compositions, and then they started to survive in an oxygen environment. But the thing is that if these organisms would be put in an oxygenic environment, they would die. So how could the transition have occurred? That's a big problem for evolutionary scenarios.
I missed a little bit of that. Do you have any, let's see. Maverick says, I'm coming from it from a theist point of view. What if those cells came from us and our decomposition after death? No. I shouldn't be dismissive. There is something important about this. Where do viruses come from?
Well, viruses, they are interdependent with life. And they cannot survive. But the thing is that life also depends on viruses. We need to have homeostasis in the environment, and who provides that are viruses. And if viruses would not have been created at the same time as life, then life could not emerge either. So that there is an interdependence here, and how that emerged, of course, that adds complexity. And that has also to be explained. And viruses, they are very complex, and they have replication mechanisms which are different from replication mechanisms in life. So this all has to be explained.
And the general view of the origin of viruses is that they came from cellular life. So there is the idea of something more complex. and the viruses were generated. But the creationist view is like what you said. But I think I understand why Maverick is asking that question. Did bacteria exist before
No,
human
after
beings?
you.
I
No,
think
after
bacteria is probably really essential for life because they do what they call nitrogen fixing.
Yeah.
Not
only fixing, but the entire nitrogen cycle, it depends on various different bacteria which compose the nitrogen cycle. And the nitrogen cycle is also interdependent with the carbon cycle and with the rare mineral cycle and with the water cycle. This is all interdependency. And that's why I am saying that the young Earth creationism viewpoint is strongly supported by that fact that these energy cycles, they are all interdependent. They are irreducible by themselves, but they are also interdependent between them. So that means that everything had to start together and all at once, and it couldn't be in a stepwise fashion.
Agreed. So I mean, that's a good question, and we'll just try to keep moving here. So, accordingly, let's see. In addition, the yeast nucleus encodes 150 mitochondrial proteins not uniquely identifiable with a single domain, but apparently eukaryotic descendants from the common ancestor. Huh. Okay. Not uniquely identifiable with a single domain, but apparently eukaryotic descendants from the common ancestor. Oh my goodness. Change Tan. She said that bacteria, archaea, like bacteria and archaea can share certain protein. I mean, bacteria and eukaryotes share certain proteins. Archaea and eukaryotes share certain proteins. So if that's the case, the yeast nucleus encodes 150 mitochondrial proteins not uniquely identifiable with a single domain, but apparently eukaryotic descendants from the common ancestor. So maybe those are orphan genes. I'm just going to move on. That's not critical, I think, at this point.
Yeah, so I'll stay another 10 minutes, but I'm tired a little bit. It's more than midnight where I am.
So maybe we'll continue the journal club another day. That's fine.
Or you can also continue by yourself, or someone else wants to come in. But I leave shortly because I'll go to bed. Okay.
So let's see. All right. We'll try to make it less than 10 minutes.
Okay.
In addition so accordingly, the yeast and human mitochondria proteomes emerged largely as products of the eukaryotic nuclear genome, 85%, and only to a lesser degree, 15% as direct descendants of endosymbiosis.
Wow, that's pure fantasy.
Ow.
Well, okay. That's an explosion of new genomic information then.
Yeah. Hey, guys. Hey, evolutionists, you want to debate eukaryotic evolution? Come on. It's like, yeah, we're going to have a debate now. Do you really want to try to engage this? I could see why you want to talk about dino bird evolution or chimp human. You guys are going to get slaughtered if you talk. So this is an open invitation. If you want to debate eukaryotic evolution, I'm game. I said I don't really like debate, but this is just getting me kind of riled up. So let's see if we have any takers. You want to take me down? Here's your chance.
Yeah.
Yeah. Oh my goodness, 85%. So let me just try to finish this paragraph, and then we'll call it a night. The strong representation of ESPs, that's eukaryotic signature proteins, those are proteins signature to the eukaryotes, in their proteomes means that mitochondria and their descendants are usefully viewed as honorary CSSs. That is, CSS is a cellular signature structure, cellular signature structure. So we're going to stop it there, but I want to point out something. So for this mitochondria here to work, 85% of the genes have to come from the nucleus. So let's even say that there was a endosymbiotic event. How does that gene migrate from here to get to here? What has to be done? Oh, it's not just a phylogenetic reconstruction that's going to solve the problem. Goodness, you have to reformat that, then relocate it. And what does the reformatting have to do? First, it has to have a nuclear export signal in the RNA, so it comes out here.
How do you orchestrate that in special by cell division where the nucleus completely disintegrates and then reassembles? And so the nuclear pores. I mean, this is an orchestration of fantastic complexity.
Yeah.
And how do you explain that emergence by evolutionary mechanism in a stepwise fashion?
So this is when, and God bless him, Brother Garte, when he was saying that it could be explained by evolution. I mean, he believes a miracle for abiogenesis. He's sympathetic for that. But I said, Sai, consider what we're talking about here. These aren't trivial steps either.
Yeah.
So we go from, so let's say magically there's a way that that gene migrated here, but now it needs all the localization signals because now it has to, an RNA has to signal it out here. It's going to get into probably here where it finds a ribosome. And now it needs a localization signal. So it doesn't go back in here or anywhere else. It has to go in here. And then now you have all the import-export signals to get it to go in here. And then
but you know, Sal, that's what I am always saying, that what has to be explained is not simply new genomic information or mutations and so forth, but in special also the signaling pathways, which are absolutely crucial. How did these signaling pathways emerge, evolve? And the languages must, I mean, I don't know if you have a little bit more information in regards of the language of the signal, of the signaling, you know, but these are epigenetic codes, which the emergence of these codes also has to be explained because in that signaling there is information being exchanged.
Yeah, I think there's just so much we don't know, but what we know today in 2021 is a lot more than we knew 20 years ago. And this is just devastating for the case of universal common ancestry. They have to invoke miracles to make it work. In all honesty, I don't debate human evolution because I think Erica had some good points for that. And it's not quite as it's more brutally obvious here. If I were debating,
I would really love to go into that a little bit more, Sal, but I think I brought up already very good points, which simply she neglects, which is the fact that the structure of the organism is principally orchestrated by the gene regulatory network, which orchestrates the genetic information when it has to be expressed. And this is something which, how do you explain that by evolutionary mechanisms? I mean, we know by the articles of Davidson that you cannot change the orchestration, otherwise everything breaks down.
Well, I really want to thank you for your time, and I promise to let you go in less than 10 minutes, and I want to honor my word. I've had a lot of fun. I hope, I tell you what, why don't you tell us about this discussion between Kunnev and Garte on your channel, and I'll try to advertise it in the video description.
It will be next Sunday, 3 is your time East time? Yeah, so it's 3 p.m. East time next Sunday, the 10th of January at my channel, The God Talk.
That's going to be exciting. And so I'm going to, I haven't put it in the video description yet for Otangelo's channel and his work. I really want to thank him. Whenever you're free, I would love to continue this. I didn't expect this to be such a fun session because Oh my goodness! When I just saw,
Yeah, I think you brought up a really interesting paper with many talking points which are stimulating to be explored. And sometimes it is you start reading a paper and suddenly something comes up which is exciting, and then you go in a completely different direction to explore something else. And it's fun because that way you are elucidating things which we still don't understand, and then suddenly something exciting we can learn.
Yeah, so if we have more of these journal clubs, we'll see if they ever appear on Speed of Sound's weather report. He's going to be busy because now that there's a Standing for Truth channel, and then this channel, and then Logical Probable, and he's fighting with Nephilim Free, he's going to be doing 2 weather reports a day.
He has also already expanded to several hours every day.
So before I say goodbye, I'd like to just acknowledge Doki Doki Bible Club. I don't know what I'd do without you. You've done so much for this channel and other channels just by being just steady. I'm going to just scroll through the names and acknowledge everyone as best as I can individually. I saw, okay, Hate Love Nothing, thank you for visiting. Stuff I watch sometimes, that was a great comment. James W., great to see you. And Doki, Honesty Angel, thank you. I miss seeing you. It's good seeing you, Honesty Angel. Daniel and the Maverick. I think I got most everyone. I hope I did. Sorry if I missed anyone. And Otangelo, thank you again for your time. And you rest up, and thank you for your hard work on this. I know you have a full-time job doing something else, and I just salute all the hard work you've devoted to what you're doing.
Yeah,
and may the Lord richly bless you for this.
Yeah, I thank you for having me. It was fun, and we'll continue this.
All righty. You take care. God bless you.
Okay. Have a nice evening. Bye-bye. See you next time. Bye.